Hello again. So the next part of this booklet, Plant Transport, is all about the transport of water and ions. So in the first part we looked at the vascular tissues and we said that there was the xylem for transporting water and ions and the phloem which transports sugar and amino acids. Both were found in vascular bundles throughout the plant. This video, because it's looking at the movement of water and ions, is going to focus mainly on the xylem and how water gets into the xylem and how it leaves the xylem, and what it does when it's in there. So we're going to break this down into three areas. The first one is the logical place to start, which is in the root, because that's where water actually enters the plant. The second part is what happens to water when it's in the stem, and the third part is the evaporation of water through the stomata, which is known as transpiration. So strap yourselves in. Now, this diagram basically just shows what's going to happen. So the first area we're going to look at is movement through the root. It involves talking about things like the apoplast and symplast pathways, the Casparin strip, and the production of root pressure. Then we'll go on to movement through the stem and through the leaves. So let's start here with water and mineral uptake by the root. So the majority of the water absorbed by the roots enters at the root hair cells, which provide a large surface area. You'll remember from GCSE that they had this shape where they had little extensions, and those were important because they increase the surface area. Now, in the first section we talked about the structure of the root, and we said that you find the xylem within the stele, which is in the center, within the center part of the root, and if water wants to enter the xylem, it has to enter the root hair cells, travel across the cortex, through the endodermis, and then into the xylem. So let's look in a bit more detail about that. So water enters through the root hair cells, which BTW is found on the epidermis. From here it must traverse the cortex and endodermis to reach the xylem. Water absorbed into the root hair cells is by osmosis, as in normal circumstances the water in the soil has a higher water potential than the root hair cells due to sugars and other solutes present in the cell. So, as we know about water potential, water will move from an area of high water potential, which in this case is in the soil, to an area of lower water potential, which is in the plant cells. So water enters the root hair cells by osmosis, and the water travels from an area of high water potential to an area of lower water potential. Now, the uptake of water is aided by the presence of a thin exchange surface. The cell surface membrane is the effective exchange surface, as the cell wall is permeable and a typical root has many thousand root hairs. Once into the root hair cell, the water moves across the cells of the cortex and into the xylem in the stele. There are two main pathways involved, as shown in this diagram below: the apoplast pathway and the symplast pathway. Now, the apoplast pathway is this one shown in red here, where water moves from the soil, and what you'll notice here about it is that it actually travels through the cell walls. Now, it will keep going until it reaches this blockade, which we'll talk about later on. But this blockade is known as the Casparin strip. So the apoplast pathway will run the whole way through the epidermis, through the cortex, but once it reaches the endodermis, where the Casparin strip is found, it has to stop. The symplast pathway is slightly different. It's represented by this brown arrow, where it goes into the cytoplasm of the cells and the water moves through the cytoplasm by osmosis. Now, the key words that you're going to link together with each pathway. So for apoplast, it's going to be that water travels through the cell walls and it travels by capillarity, which we will talk about later. The symplast pathway is the movement of water through the cytoplasm of each cells, and it occurs due to osmosis. so the setting up of a water potential gradient, and then water moving from an area of higher water potential to lower water potential. Now this diagram also shows the apoplast and symplast pathways. So you can see the apoplast pathway is the movement of water through the cell wall, and the symplast pathway is movement through the cytoplasm. So let's look at each one in a bit more detail. First of all then, we've got the apoplast pathway. Now this is when water moves through the cell walls in the spaces between the cellulose microfibrils. So this here is basically a cross section of a plant cell wall, and the cellulose fibrils are pretty much arranged like that, where they crisscross over each other, so the water can actually move through these little gaps between them by capillarity. Now, it moves by capillarity because of the cohesive properties of water. Now this basically means that water molecules will stick to each other because of hydrogen bonding. Those of you who maybe do chemistry might have looked at this, where you look at the structure of water molecules, and what you'll have seen is that water molecules will be attracted to each other because of hydrogen bonding, which is pretty much between the slightly negative oxygens and the slightly positive hydrogens. So water molecules are very good at sticking together. So basically what happens is, if water molecules enter the apoplast pathway, and this is a little water molecule, they'll move along, and the ones behind it will be attracted to it, so they'll stick together and they'll be pulled along. Now you can actually see this in real life. This little experiment on the right hand side is a real life demonstration of capillarity. Basically you could do this at home, where if you get a piece of kitchen roll, 2 beakers of water or 2 cups of water, and in one of them you start off with an empty cup, and in the other one you start off with a full cup, the water will actually move very slowly by capillarity from one cup to the next. And that's basically because the water molecules get absorbed into this, but as they move up, the water molecules behind it, because of capillarity, stick to it and they get pulled along with it. Most of the water will move across the route in this way because there is a low resistance for the movement of water in the apoplast pathway, so it's faster and more water can move that way. The symplast pathway is slightly different. In this, water moves by osmosis from cell cytoplasm to cell cytoplasm via the plasmodesmata, which we talked about in the previous video, and we said the plasmodesmata were little strands of cytoplasm which link 2 adjacent cells together. So the water simply moves between the cytoplasms via the plasmodesmata. The water moves based on water potential. So for example, in the root hair cell takes in water, its water potential increases, becomes less negative. So this has a higher water potential than adjacent cells, so the water moves into the adjacent cells. In other words, it's continuous until it reaches the xylem, so a water potential gradient is set up between the soil and the xylem. Water then leaves the xylem and travels to the leaves in a similar way. Now, just on that little part, so if for example we have the soil here, this has got a high water potential. This is the first cell of the epidermis, so this could be a root hair cell. This would have a low water potential, and that's because there's solutes and things dissolved in the cell. So the water is going to move from an area of high water potential to low water potential across the selectively permeable membrane. But what happens next is, once this cell has taken in that water, its water potential becomes higher or less negative. So that means that now this epidermal cell has got water which it can pass on to this cell, which still has a low water potential. So the water moves from here to here and then from here to here via the plasmodesmata. Again, this cell takes in water, so now it has a higher water potential, so it can pass it on to this cell. And that keeps going the whole way along until we reach the xylem, and then something slightly different happens, which we'll talk about. But this is what I mean by setting up a water potential gradient. Here is where we have the highest water potential. Here is where we have the lowest water potential. So we've set up a gradient the whole way from there to there, which is what I mean by this point. Now, it's important to understand as well, though, we were talking mostly about water there with the Applepass and Simplast pathways, but the ions then can enter the root hair cells by facilitated diffusion or by active transport, depending on the concentration gradient for the particular ion that we're talking about. Intake of ions is often by active transport, causing the ions to build up within the cells, thereby requiring the ions to be moved against the concentration gradient. And if you think back to GCSE in ecology, I know, ecology, right? But you had this little diagram of a root hair cell, and around the outside you had little nitrate ions, and within the root hair cell you had a really high concentration of nitrate ions. Now remember, the nitrates were used for growth, and this is where we talked about the nitrate ions moving from a low concentration to a high concentration so that the plant cells could store these nitrates and then use them for growth. So quite often, root hair cells will use active transport to try and get as many ions out of the soil as they possibly can. Then once they're in the root, they'll be dissolved in water and they can be transported through the xylem. Now this little diagram just shows you the Applepass-Simplast pathways and also the xylem. So let's do a little bit of colouring in. So here we've got the xylem vessel over here. Here we have got the epidermis, which is composed of the root hair cells. We actually don't need to know about this little layer here, so let's just assume that it's part of the epidermis. Then we've got the cortex. Now, parenchyma is just a word used to describe packing cells. We don't need to know that. So this layer here is the cortex, which remember we said was just undifferentiated cells. So here we've got the cortex. And then we have this little layer, which is called we'll go for this color. This little layer, which is the endodermis. And the endodermis is where you find this Casparin strip. Now, the purpose of this diagram is to show you the Applepass and Simplast pathways and show you where the Casparin strip is found in relation to the other structures. So out here, we have got all the soil particles. So these big circles here are soil particles or soil crumbs. We have water around them. This is a root hair cell. Now, what we can see here is that the water moves in to the root hair cell and travels this way through the cytoplasms of the cells. And that is the Simplast pathway. The other pathway we can see, I'll draw it in red here because it's not shown for you at the minute, is the Applepass pathway, where the water moves between the microfibrils of the cell wall. Now, watch very carefully at this point. The Applepass pathway can only go to the endodermis, and the Casparin strip is there because it actually stops the Applepass pathway. It diverts water into the Simplast pathway. So I'll maybe use a less chunky pen for that. So the Applepass pathway stops at the Casparin strip, and the Casparin strip is found in the endodermis, and it is there to block the Applepass pathway. I'll find out why in a minute. So the Casparin strip diverts the water from the Applepass pathway to the Simplast pathway. So the Applepass is shown in red. Now, maybe if we get rid of all those colours, I don't know, maybe that's easier for you to see. So the water, once it's in the Simplast pathway, can then get transferred into the xylem. And this section is going to describe how that actually happens. Now just linking that to our root structure, the root hair cells were on the outside, and for water to get from the soil to the xylem, it has to travel across the root hairs in the epidermis, through the cortex, through the endodermis, and then into the xylem. Now remember, our wee Casparin strip is found within the endodermis. So where in the root is the Casparin strip located? What is it composed of, and what is its significance with respect to the movement of water from the cortex into the xylem? Now this bit's pretty important, and there's some important terminology here. So the Casparin strip is a waterproof layer made up of suberin, which is a waxy substance, and you'll know wax is waterproof, embedded in the cell walls of the endodermal cells of the root. So the Casparin strip is found in the endodermal cells, and it's made up of suberin. It diverts water away from the apoplast pathway and forces it into the Simplast pathway. Now, this is the key words. It's important because it ensures all water is under metabolic control of the plant. And it means that the endodermal cells pump the mineral ions into the xylem using energy from ATP, which lowers the water potential in the xylem compared to the endodermal cell, which creates a water potential gradient that draws water from the endodermal cells into the xylem. Now, this water, which pushes upwards, is root pressure. So the question is, what is root pressure? Root pressure is the movement of water along a potential gradient into the bottom of the xylem, which creates a root pressure force that helps move water up the plant. Now, in large plants, for example trees, the force generated by root pressure is not enough to move water up the entire plant, but in a very small plant, it may be the main way which moves water. Now, if you think about this, some trees can be up to, let's say, 30 meters. So that little push at the bottom in the roots isn't enough to move it over that distance. It must be something else moving it over that distance. But in a very small plant, like if you think of something like a small cactus, which might only be a few millimeters, root pressure would actually be enough to draw it the whole way up the plant. And if you look at this, these are some redwoods that you would find in parts of North America, which can grow up to, I think, 50 or 60 meters tall, maybe even taller than that. So the main way that they actually draw water up is from the pull at the top of the tree, the transpirational pull. So as we've said then, the movement of water involves three parts: the movement through the roots, movement up through the stem and the xylem, and movement through the leaves. Okay, so what we didn't say in the first part is that some people like to think of this as a push at the roots, which is the root pressure, and then a pull at the top, which is created by transpiration. And those two forces acting together basically get water into the xylem, and then the pull helps pull it up through the stem and out through the leaves. So the next little area we're about to look at is movement through the xylem. So we have got the water into the xylem because it was pumped in there. Now let's look at how it actually gets up through the xylem. So when we're looking at movement through the xylem, there's two very important forces here which are going to help us out, and it's adhesion and cohesion. So we talked about cohesion previously. Cohesion was when the water molecules stick together because of hydrogen bonding. Adhesion basically means that the water molecules can stick to the wall of the xylem. Now that's important because if you think about it, water is moving upwards through the plant, so it has to overcome the force of gravity. So it's really important that the water molecules will actually stick to the walls of the xylem, which makes it easier to overcome the gravitational pull. So this section then, transport of water up through the root and stem in the xylem. In most plants, the movement of water and ions from root hair to xylem involves the distance of a few millimeters or centimeters at the most. Movement up the xylem in the root and stem can be meters though. As water evaporates out of the stomata in the leaf, it creates a negative pressure. So we said that negative pressure was pretty much a suction. So that negative pressure pulls the water column up through the xylem as a mass flow movement. This process requires the water column to form a continuous, unbroken pathway through the xylem, known as the transpiration stream. Water molecules form hydrogen bonds between one another, known as cohesion, and this sticks the molecules together. It is the cohesive properties of water that allows the water to be sucked up the xylem in a continuous column as the water at the edge of the column evaporates out of the liquid. Now, this continuous water column is actually so important. Say somebody buys you a nice wee bunch of flowers. Oh, cute. Maybe from your wee boyfriend or something. Somebody buys you a nice wee bunch of flowers. You take them and you cut them. It's really important that you cut them underwater, because if you don't cut them underwater, then you get an air bubble within the stream, and that means that water can't be pulled up properly, so your flowers will die sooner. And that's the same as plants in the wild. They need a continuous, unbroken pathway of water through the xylem so that they can basically transport water throughout all the different parts of the plant. Now, this is known as the cohesion tension theory, and the driving force is the evaporation or transpiration out of the leaves, resulting in a transpirational pull. So the cohesion, it's called this because the cohesion is the water molecules sticking together, and the tension comes from the transpirational pull, or the negative pressure. Now, as we said in the previous section, root pressure was a pretty low force, whereas transpiration creates this negative pressure, and it's the driving force behind the water moving through the plant. Now, the evidence for the cohesion tension theory then is this. If the water column in a xylem vessel is broken and an air gap appears, water below it cannot be pulled up. So if you get a broken water column, the water below it will not be pulled up. During the day when transpiration is normally at its greatest, there is much more tension or negative pressure in the xylem. The negative pressure tends to pull the wall of the trunks or pull the wall of the xylem vessels in and can reduce the diameter of a tree, which is actually what we're looking at here. So this shows you how at different times of the day, the diameter of the tree, which is this line, will decrease or change in relation to the rate of transpiration. So if you look at this part, this is where the rate of transpiration is quite low between here and here, probably because it's dark, so the stomata would be closed. So therefore, you have a wide diameter. But then here, around this time is where the sun comes up, so there's more light, so the plant starts photosynthesizing, so the stomata open, and that means that because at this point the stomata begin to open, that means there's more transpiration. So there is the negative pressure created, and that negative pressure pulls the water up through the xylem, but it also pulls in the sides of the xylem vessel. Now, luckily for the xylem, it contains lignin, which helps prevent it collapsing completely, but it still does get squeezed a little bit. Now, when we reach this time, between, let's say, 12 to 2 o'clock, where the sun's going to be highest in the sky, there'll be most light available, probably also coincides with the highest temperatures. That's where the rate of transpiration is going to be highest. And that also coincides with where the diameter of the trunk is going to be smallest, because at this point there's the greatest transpirational pull. So if this is the xylem, where the transpiration is pulling water up, it squeezes in the walls of the xylem. And if you think about it, that will have a collective effect, because if this is the branch of a tree, there will be probably thousands of little xylem vessels which all get compressed in at the same time. Then later on, as there's less light or in lower temperatures, the stomata start to close, so the rate of transpiration drops. Now, as we've said, the cohesion tension theory is probably the most important process involved in moving water up through the xylem and the roots and stems. Root pressure is also important in some plants, but plays a small role in all species. Final part then is what is adhesion and how do the adhesive properties of water contribute to its movement of water through. So adhesion then, well, the water molecules must move upwards through the xylem against the force of gravity. So adhesion means that the water molecules will stick to other materials. So in this case, it sticks to the walls of the xylem. Now, that doesn't necessarily move the water upwards, but it greatly reduces the forces needed to create the transpirational pull, and it helps move the water against the force of gravity. This section then is the final section. So we've got water into the roots and in the xylem. We've brought it up through the xylem because of the cohesion tension theory, and now this is what creates the transpirational pull. So the transport of water through the leaf and the evaporation of water from the leaf. Water enters the leaf in the midrib, which is the vascular bundle, the main vascular bundle. In most leaves, this midrib splits into a number of veins that will distribute water across the leaf. So we've got our leaf, the midrib going up the centre, and the veins going off to different parts of the leaf. Water passes from the vein to the surrounding cells, where some is used in photosynthesis or providing turgor, but most is lost in transpiration. This is due to water evaporating from the cell surface membranes of the spongy mesophyll cells into the air spaces, with the water vapour diffusing down the concentration gradient and out of the stomata, and that is transpiration. So all together, this part here, water evaporating from the cell surface membranes of the spongy mesophyll cells into the air spaces with water diffusing down the concentration gradient and out of the stomata, that's your definition for transpiration. This process sets up the water potential gradient that is ultimately responsible for the transpirational pull. Now the water will travel across the leaf from the xylem to the spongy mesophyll cells by the same processes involved in water transport in the root, by the apoplast and symplast pathways. So as I say, transpiration is the evaporation of water molecules from cell surface membranes of the mesophyll cells into the air spaces and diffusion down the water potential gradient out into the surrounding atmosphere. And remember, that is what creates the negative pressure which drives the cohesion tension theory. Now, water loss through the stomata is known as stomatal transpiration, and this just explains what we were talking about. So here we have part of a mesophytic leaf, here we have the xylem, here we have the mesophyll cells, here we have the air space, and here's the stomata. So this is shown here, where we've got the xylem. Now you'll notice here with those, there's the continuous column of water, because of the broken down end cell walls of the xylem vessels. The water moves up in a continuous stream out through the pits to the surrounding cells, and then it evaporates from the mesophyll cells into the air space and then out the stomata, down the water potential gradient. Now, this little diagram just ties everything we've looked at together. So we've got the first area, which was moving water into the xylem from the soil. So movement of water there across the root and into the xylem. The second area was moving water up through the xylem because of the cohesion tension theory. And the third area was the movement of water through the xylem, out into the leaf, and then evaporation from the cells, and then diffusion out through the stomata. And that area was transpiration, creating the negative pressure, which basically drives this whole process. So let's have a look at it. So down here in the root, main things that we talked about was the apoplast and the symplast pathways. So there was the apoplast. Here's the symplast. Now the difference was the apoplast moves water molecules through the cell walls, and that's where most water moves. The symplast pathway moves water by osmosis through the cell cytoplasms, but the apoplast pathway was blocked by the Casparin strip, which is found in the endodermis. When the water reaches the Casparin strip, it gets diverted into the symplast pathway. Now, how we get water from here to here was very simple. As the water is moving across the route, it takes with it ions. The ions get pumped across into the xylem, and that creates a lower water potential. So it sets up a gradient from here the whole way across the route right to there. Because there's a water potential gradient, the water molecules then move into the xylem. When the water moves in here to follow the ions, it creates this root pressure, which forces the water upwards through the xylem. Now the water molecules move upwards very easily because of their cohesive properties, which basically means that one water molecule at the front will pull the one behind it because of hydrogen bonding. As they move up, the water molecules will also stick to the walls of the xylem because of their adhesive properties. And as well as that, we have the transpirational pull, which pulls the water molecules up. Now it's really important that you've got a continuous column there, because if the continuous column gets broken, the hydrogen bonds get broken and then the water will not get pulled up. So we need a continuous stream of water. The water molecules move up in the xylem and they will branch off to little leaves in the midrib. The midrib will then branch off into the little veins, and the water will move out through the pits. Now when it moves out through the pits, it can either travel by the apoplast pathway or it can travel by the symplast pathway. So the same as down in the root. When the water reaches here, it will evaporate from the cell surface membranes of the mesophyll cells into the stomatal chamber, because there's a water potential gradient where we have a high water potential there, low water potential there, and an even lower water potential out here. So it sets up a water potential gradient from here to here. And this transpirational pull pulls water the whole way up through the plant from way back down here. And that's the whole summary of the movement of water through plants. Yay! Thank you. Hope you had fun. Goodbye.